Thanks for the introduction, and thanks for the opportunity to speak to the Demystifying Medicine program. And anyone? There we are. So the poster that we did have for this talk did have 4 objectives, and I'm going to try to speak to the last 2 of these objectives. I'm not the engineer, and I'm not a microfluidics expert, and that's why I've actually partnered with Ravi, and we've got an ongoing relationship with that Sick Kids grant that we did talk about, and I'll try to get into some of that data a little bit later. But first thing what I want to do is kind of take a high-level approach on like why are we going into all of this research with microfluidics and new technology? Why don't we just take blood or take some cells and grow them in a dish like we historically have? And so I wanted to highlight the challenges. I'm a PhD, but I'm not an MD. I don't see patients, but I do interact with physicians that do see patients, and I hear about the challenges that they face and the meetings that I go to, and I hear about the challenges about, this room is clearly diverse, but yet we treat everyone very similarly when it comes to their given condition. And as I say, you can cast a broad stroke here. The audience is quite diverse, and clearly, all of us are humans in this room, but our genes are quite different. And those genes, let's just say they're a foundation for how we operate and how we can respond to various, whether it's a drug treatment, whether we go on to develop chronic disease, or whether we have a lung condition. Our genes may be the foundation for that. They're not going to be necessarily, just because you have that gene, it might not lead to the disease. It may, but there might be some other factors. And indeed, our environments are also different, and this is where the environment and genes can interact. I do some air pollution work as well, and I'm interested in how the air we breathe can impact lung health and disease. And this is a satellite map of particulate matter of a certain size throughout the world. And what you can see is in the red regions, and I'll highlight China here, which we know can tend to be pretty high. Throughout Africa, this could probably be attributable to some of the dust in the deserts. But this is air pollution. And if you actually take me and you put me, the same genes, in this different environment, I will respond differently. I will have a different response to therapy. I may go on to develop a disease that I didn't have in a cleaner environment. So it's not just the genes, but it's the environment that can actually influence us and how we can go on to develop conditions. Our lifestyles can also be very different. And I'm showing you past and present here. In my younger days, and this was at a bachelor party, I sort of unearthed some unflattering photos of me, and this is what I used to do. This is what I used to do and have a good time. But that lifestyle I've left behind me, hopefully, and now I try to be a little bit more active and take care of myself. But I'm the same genes, but the lifestyle is different, and that lifestyle and that exposure is going to impact my response maybe to therapy, or again, ability to develop a chronic lung disease. And I think really when you put that together with lifestyle and environment alongside genes, this really can come together in this striking photo, and I don't know if anybody has seen this photo. It's a real photo. I give you the reference beneath from the New England Journal. This is an Australian truck driver, 27, 28 years of experience driving a truck, did not wear sunscreen. So his one side of his face that was always getting hit with sun, maybe because it was a morning route, and that's what got tanned, and that's what got burned, and that got this UV exposure. He didn't have a stroke. This is just repeated exposure to sun on the one side of his face over the course of his career. And so there's his environment that he's in, this harsh UV environment, his lifestyle. He didn't really look after his skin. He didn't have protection for his skin. And then that impacted some pathology and susceptibility with his genes. So, we operate in a post-truth environment right now, unfortunately, but I'm going to try to lay down some truths that we have. Most of us start off with different genes unless we're identical twins. The genes that we do have, that's a foundation, but they can be impacted by our environments as well as our lifestyles. And this is really one thing that I feel it gets overlooked in the gene-environment interaction field. Every individual is unique, and because of these gene-environment interactions, but that also has a temporal feature to it, so that's dynamic. I gave you an example that, yes, I used to be this individual that would have a couple of drinks, and then now I was this individual running. That's a dynamic environment. And so therefore, at different stages in our lives, we may respond differently to therapies. And that can have important clinical implications. So the problem is, this is the clinical problem, is how do we treat each person at each point in their life optimally? Does it make financial sense? Can we really do that with everyone in this room that is different? Should we tailor doses or treatment strategies uniquely to each individual? Can we do that? And can we do that in a safe and effective manner? And one of the solutions gets a lot of hype, and I'm sure there's been a Demystifying Medicine talk on precision medicine or personalized medicine, but I think this strategy holds hope. It's how we pursue the strategy, but it does have hope. And this precision medicine strategy, and this is the logo for the NIH, which was an Obama initiative, it's an approach where we try to incorporate the genes of the individual, their environment that they live in, and the lifestyle that they lead, and how can that influence their health and the development of disease. So that's the NIH. Closer to home, in Canada, we have a precision medicine focus within the Canadian Institutes of Health Research, which funds medical research in Canada. It's a little bit more verbose here, but the same message comes out, is that yes, we feel that using various omics technologies, genomics, epigenomics, and proteomics, maybe we can develop rapid diagnostics, as Ravi was saying, lower volumes. We can do more experiments in less time with less resources using nano or micro technology. Can we create diagnostics for these individuals? Instead of just doing one test that takes 2 weeks, can we do 10,000 tests that take a day? And really, that is a focus of the CHR, and this is sort of where some of our, Ravi and my work comes together. And I just wanted to highlight that within this CHR theme, there is nanotechnology, molecular diagnostics, precision diagnostics, and things that Ravi's technology can definitely feed into. So we've got this problem, how do we treat each person individually and optimally? We've got this solution, precision medicine, nice term, but what does it actually mean? So I would say, when I think about it, okay, you need to make some assumptions about precision medicine. You can't do everything. So I took a step back and said, what is it that I can do to assess precision medicine in lung health and disease? And my assumption was, okay, I need an easily accessible tissue. I need it to be clinically relevant and related to the disease that I'm looking at. I say cell or tissue sample, whatever I can get my hands on. And it needs to have a functional response. My bias, and I'll admit it, is if I just see a gene, that the gene's there, that's one thing, but it's not giving me function. I need to see that it's either working normally or working abnormally, and then I'll know that that's a potential target or leading to the individual's disease. So these are just my assumptions. They're not necessarily the truth, but this is just what I'm using to base my research on. So the introduction said that I was into respiratory mucosal immunology. Really narrowing that down, I look at epithelial cells in the airways, which is the first line of defense in our lungs and in our upper airways in our nose. And they're easily accessible. This is a fairly routine procedure. You can do this on an awake individual. You can get a flexible bronchoscope that has a little brush, kind of like a pipe cleaner. You do a little brush down here in the bronch, and we can isolate epithelial cells. Some people can do it without any anesthetic. Some people need a little bit of lidocaine so that they don't cough and gag, but other than that, it's a fairly non-invasive procedure. If you want to go even more non-invasive, you can go to the upper airways, and you can this is a nasal swab, but if you swap it out with a brush, you can just go into the nose, into the sinus, and get epithelial cells. So as far as my assumptions go, or my requirements, epithelial cells are easily accessible. Are they clinically relevant in lung health and disease? I will make the case that yes, indeed, they are relevant. In asthma, for example, our dean in the Faculty of Health Sciences, Dr. Paul Byrne, has been pioneering some clinical trials that are testing novel TSLP, thymic stromal lymphopoietin antibodies, inhibitor antibodies, and they've been showing promise in asthma. TSLP comes from epithelial cells. So you see this link. There's an epithelial cell. Okay, it's producing a molecule that's important in asthma. Dr. O'Byrne's group has shown that if you block that molecule, you can help alleviate some of the symptoms of asthma. So it's definitely clinically relevant in asthma. In COPD, which is a disease that individuals that smoke for over the course of their lifetime may develop, and as well, when we take samples from individuals that have smoked that do not develop COPD and compare that to individuals that smoke that do develop COPD, there can be a signature in the airway epithelium that relates to disease severity. So again, a bit of a biomarker for disease severity, so maybe we can look at responders or non-responders or people that are going to go on to develop disease, and maybe we can intervene earlier in individuals that smoke. Lastly, this is a genetic disease, and this is sort of one of those, you either have it or you don't, so it's actually built into your foundation, into your genes. If you're unfortunate to have one of 2,000 variants in the CFTR gene, this may lead to reduced function of the CFTR protein, which can lead to dehydration of your airways and a sticky mucus, which leads to recurrent infections and loss in lung function. So that's an epithelium abnormality. That gene is expressed in the airway epithelium. So these are three diseases, and I gave you 3 examples to say that airway epithelial cells are clinically relevant for lung diseases, and they are also easily accessible. So now, my last strategy, I said if we wanted to go down the precision medicine route, is okay, we've got the cells, we can get them, they seem to be involved in the pathology of the disease, or maybe they can give us insight into how someone might respond to therapy. Is there a functional response that I can measure from these cells? And that's a segue into this second objective that I had, which is to provide the functional example of precision medicine in cystic fibrosis. And this is work that I've done with people down the road at Sick Kids, and that we're continuing on with Ravi. So I need to get into a little bit more at the granular level of some of the detail of the research that we do. We look at airway epithelial cells, and as was mentioned, I'm interested in these proteins that are called ABC transporters. This is just an image of the ABC transporter, and as the name intends, it transports things from the inside of the cell to the outside of the cell. One of those molecules that it transports is cyclic AMP. It can do some other things, but I'm only going to focus on cyclic AMP. Within the plasma membrane of an epithelial cell, in close proximity, is CFTR, and this is that gene that is, there are variants of that can lead to cystic fibrosis. And CFTR is also a transporter. It's actually an ABC transporter as well, and it transports chloride out of the cell. Now, in the individuals that do not have cystic fibrosis, chloride will go out of the cell, and then that'll lead to water going out of the cell, and that'll lead to hydration of the airway surface lining fluid and normal mucus. Whereas if this isn't working properly, then what happens is the mucus, chloride doesn't go out, water doesn't go out, and the mucus becomes dehydrated and sticky. I've drawn a red P on here because this is a phosphorylation site which influences the activity of this transporter. So I say that these are both ABC transporters, so they're linked actually genetically because they are derived, they believe to be derived from each other over evolution. But they're also linked within the cell membrane physically. And here's a scaffolding protein that links these two proteins together, ABCC4 and CFTR. And what I've said to you without showing the data is that ABCC4 transports cyclic AMP outside of the cell. And I'm going to show you why that's important in the context of cystic fibrosis. So this is just some data where I've been working on developing some novel ABCC4 inhibitors. So I'm just calling it ABCC4 inhibitor because of confidentiality reasons, and I'm not showing you any structure. But what we're able to do is we have an assay that measures the amount of cyclic AMP that goes outside of an epithelial cell. And so that goes out of the cell, But then what I have here on the x-axis is increasing concentrations of my candidate compound. This is supposed to block ABCC4, and it's supposed to block extracellular CAMP transport. That's why you see as we increase the dose, there's less getting out of the cell because the transporter is being blocked. So I schematically show that here, ABCC4 inhibitors, they can come and they can block this. CAMP disappears, and what happens is CAMP, I'm not showing you all the data, but CAMP concentrations increase inside the cell. A downstream consequence of that is protein kinase A activation, which is a kinase which can phosphorylate CFTR. And as I described, CFTR, when it's phosphorylated, it works better. So hopefully you can see a little bit of the connection here. I block here, I can make this work better. That's the concept. But we needed to test that. And we needed to test that in real individuals or samples from real individuals that had cystic fibrosis. And this is where the collaboration with Sick Kids Hospital down the road in Toronto happened. And so what we were able to do is we were able to take those nasal swabs. So very easily, we get a little swab in here. We take those cells, we filter them out. We treat them with antibiotics to try to kill any bugs that may be in the nose. And then we can grow those cells. We grow those cells on transwells and we feed them from underneath, and they're exposed to air on the top. Very similar to what's happening in your nose or in your lungs. Now we can take those cells and we can either treat them with a vehicle, control, or we can treat them with the drug. And this assay that we develop is a fluorescence-based assay. More fluorescence means more CFTR activity, which is a good thing. So this is the punchline in the paper that we published last summer, and I will take some time to go through it so that you can really see the consequences of the data. This is the measure of CFTR activity. So this is what you want more of. The white bars are healthy individuals that don't have any variants in CFTR. So this is sort of like for individuals in this room that do not have cystic fibrosis, this is how we would measure up. Now these are three different individuals, so this is where the precision medicine comes in. Three different individuals that have the same homozygous mutation in their CFTR gene. This represents 80% of the CF population. Underneath the x-axis, we have the combinations of drugs that are out there. Currently, the individuals that have delta F508 homozygous deletions, they're given clinically, this is what's approved, lumacaftor and ivacaftor, and that's a $100,000 a year drug for these individuals. And so what you can see in these three individuals, the light gray bar to the first dark bar, that's the clinical drug that's being used, and they're probably getting a doubling of their signal, but it's nowhere near the non-cystic fibrosis patient samples. So they're getting better, but they're not getting back to normal. Unfortunately, in these three different patients that had the delta F508 variant, when we added my ABCC4 blocker, we didn't see any improved benefit. So this was a little bit disappointing because the concept seemed to be falling apart. But there was some data that we had that suggested that it could work, and we pursued this idea a little bit further. And what we were able to see, I told you there's 2,000 variants in CFTR. Well, we decided to look at some of the other variants. And when we looked at these other variants that make up 5 to 10% of the CF population, the G551D variant, now you're comparing the red bar to the blue bar. Because for these people, at $100,000 a year as well, they only get the blue drug. But then when we add my ABCC4 inhibitor on top of it, we're doubling that. And we're bringing it much closer to the wild type and definitely higher than the other delta F508 patients. So this is an example of showing function. These are functionally relevant. They're cells that actually are leading to the abnormality in these individuals, and we're using drugs that are clinically relevant, and we're boosting that response at a precision level in these individuals. So this is the assay that I described. It's a conventional plate-based assay. It takes some time to grow these cells. And as Ravi was saying, time is money, and if we can do this faster, smaller, cheaper, then maybe we can help more people and scale this technology. And that's really where the Sick Kids grant comes into play, using Ravi's technology as well as Christine Baer's technology from down the road. We're going to try to scale this down into an organ-on-a-chip device where we can have air on the top, media on the bottom. We can have multiple channels. This is just an example where we have the apical channel on the top and a basal channel on the bottom where we can feed from the bottom or perfuse drug through on the bottom. You can imagine that this could exist in parallel. So if clinically available, there's 20 combinations of drugs that we can give to any given cystic fibrosis patient. Well, let's grow that individual cells and let's perfuse through all 20 different combinations and find out the combination of drugs that works best for that patient. Not for you and average them together, but let's find it at an individual level, and we could do that over time if needed. We could look at them 6 months down the road, a year down the road, 5 years down the road, see if they're progressing, see if they need to modify their therapy. As well, we've been working with a company out west as well as Ravi has, and this is a pioneer in 3D tissue printing. So maybe we can also merge 3D tissue printing with that microfluidic technology. And this is a biotech company out in Vancouver, and they're doing 3D tissue printing. Here's them creating what is like an airway structure, a tube structure. And I suggest to you guys, if you guys are interested in this technology, to look into this company. So with that, I hope I've met my two objectives here, and that's to recognize the challenge that on an individual level, and this is just lung disease because that's what I'm interested in, but hopefully you can appreciate that this applies across all diseases and all situations where genes are interacting with the environment and our lifestyle. It's very difficult to tailor the therapy, but maybe with the application of microfluidics technologies and tissue printing and tissue engineering, we can really take this down to a precision level, at a cost-effective level, to optimize care for all the patients that need it. So with that, I will end my talk and take any questions or comments.